your world right now 2 major results, aren't you? So perhaps you could share what those are, and they're relevant obviously to our conversation this afternoon.
Absolutely. So I'll be presenting the MajesTEC-3 subgroup analyses of high-risk patients. So as all of you are well aware, TEC-DARA, so teclistamab plus daratumumab, showed amazing results in MajesTEC-3. Dr. Mateos got a standing ovation at ASH for it, approved in the US already, hopefully in Europe any moment now. Historically, there are some patients who don't do as well with any kind of treatment in the relapse setting versus others. And I think cytogenetic high risk comes to mind. What I was really interested in is these functional high risk patients, right? Patients with early relapse where, for whatever reason, something about their disease biology spooks you. Spooks the patient, it spooks you as a doctor. They got their transplant and less than a year later, they're relapsing. What's going on? Historically, those patients have done terribly with treatments thereafter. And I think basically what we'll present here is if you look at the TEC-DARA subgroup data of patients who had functional high-risk disease, so meaning 1 prior line of treatment and only 18 or fewer months of remission, either after a starting frontline treatment or transplant, and went on to the study, as an example, in the patients in this large phase 3 study who got DARA-PD or DARA-VD, 3-year PFS was 0%. Nobody made it to the 3-year mark of these functional high-risk patients because, again, they do so poorly with every other line of treatment. With TEC-DARA, it was 77% 3-year PFS, right? Which I think is just remarkable that you're starting to close that gap finally. Not 100%, we have work to do, and that's where I think some of these cell models will be helpful. At the plenary session here at EHA, Dr. Pete Voorhees from North Carolina will be presenting the MonumenTAL-3 data. So that's a little bit different, and this is not a cell mod and also not BCMA. This was talquetamab, a GPRC5D bispecific, plus daratumumab, plus or minus pomalidomide. So there was a tal-dara-pom and a tal-dara arm. Both were separately compared against DARA-PD. And in brief, we also saw substantially significant PFS benefits and overall survival benefit as well. Quality of life needs to be preserved. So I think in the future, early relapse myeloma will be even more confusing. We've already talked about CAR-T, TEC-DARA, MEZI-KD. Now we also will have talquetamab-based combinations as well. The takeaway pertinent to this discussion, we already talked about this, there are patients who will prefer MEZI-KD because they're worried about other side effects, they want a more oral-based regimen, they're worried about infections. What's interesting, and Dr. Voorhees will touch on this, Pete will touch on this, the study was not designed to compare tal-dara versus tal-dara-pom, but as you alluded to, in our head, this is the first thing we do is to kind of look at these arms. The POM offered perhaps a little bit of a benefit if you squint at the curves just right, but much more fatigue, much more cytopenias, the brain fog, a lot of these side effects of pomalidomide just cause issues. And honestly, I had patients on this study, and these are patients who used to handle POM 4 milligrams easily. Now POM 2 milligrams, they were just struggling with. And it may be something about the pomalidomide in this setting, the best of POM and the worst of POM come out at the same time. As you alluded to, mezigdomide and Iber don't have as many of those off-target side effects, so I'm hopeful that in the future, as you alluded to, Thanos, when you see trispecifics plus Iber or Mezi or talquetamab plus Iber or Mezi, hopefully you'll get the benefit of that synergy and hopefully fewer of these non-hematologic side effects.
No, thank you, Rahul. It's extremely well said. I think the important point is less it becomes more, and the issue then is that quality of life and outcome is dramatically improved. Well, I think we can say that this has been an extraordinary, productive and sort of fruitful series of meetings at ASCO and EHA, and I personally think that for having been in the myeloma research space for some time now, it's dizzying, isn't it? Or dizzying and dazzling, I guess, the number of opportunities we have. But I have to say, I think amongst